Hi friends, you're back with me, Professor Girish Kukreja. And today, while performing many experiments, we need to find out the concentration of a particular compound. And then most commonly used method in a general microbiology, biotechnology or a biochemistry laboratory is spectrophotometry. A similar kind of case was encountered here where the concentration was to be found out. You are very much bugged up with this milligram per ml and microgram per ml, but then I know that you're all well versed with it, right? At least let us assume that we are because it is not that complicated, right? So it was a tyrosinase solution which was given to me. I'm so lucky I'm not in the laboratory right now, but I know that you are also equally lucky because it is because of the people who are working in the laboratory that we are getting so many new things coming up. Great. So tyrosinase solution which has a absorption at 280 nanometer of 0.50. Yeah. So I had a solution of tyrosinase which was I don't know like of what concentration, but when I took its absorption at 280 nanometer, it gave me a reading of 0.50. And I want to find out okay, what exactly is the concentration of that, and that too in the units of milligram per ml and microgram per ml. Like how will you go about it, right? For this, one more thing we should know, like from a pre-information, that the absorption coefficient, what you call as your epsilon E for 1% at 280 nanometer for tyrosinase is around 24.9. So this means that if I am having a 1% solution of tyrosinase and I am taking the absorption at 280 nanometer in the cuvette of 1 centimeter path length, then its absorption is 24.9, right? So with this Beer's and Lambert's law, we know that A is equal to epsilon CL, right? Go back for this derivations and some common questions asked in Viva on some other video on the same channel. So you just search Girish Kukreja, Beer's and Lambert's law, microbiology, and then you'll get to know about this. Anyways, so here I have a better option, like using this you can go for C1 upon A1 which is equal to C2 upon A2, right? You know the simple problems which you used to solve in when you are in lower standards, like if one apple is for this much, then 10 apples are for how much, right? So similar kind of a thing here we have. So we have like concentration C1, the standard concentration which we know, that is 1%, right? So 1% concentration. So here I'll take 1, right? Absorption of that is around 24.9. Great. C2, I don't know. I don't know, therefore I'm doing it. And then I want to, no, I don't want to find out. I have the absorption of 0.50. I know you remember when you were kids, you perform this cross multiply. Let's do that. Things are very easy. Let's not make it complicated, right? So I have the concentration now at my hand. It is C2, which will be around 0.50 upon 24.9. Please solve it for me. I am weak in mathematics. Somewhere, like considering this as around 25, this will be around 0.02, right? So I have the concentration of 0.02%. I'm going to be very raw this time. I'm not going to give you some tricks and formulas. Let us be simple sometimes, right? So it is 0.02%. So it is 0.02 grams in your 100 ml. Without going much into detail, like I want to find it out in milligrams. So let me find out how much milligrams is it multiplied by 1000, right? So it is 20 milligrams in 100 ml, and I want to find it per ml. So let me divide by 100. So it is 0.2 milligram per ml, as simple as it is. I have it, and now I want to find it in microgram. It's simple, yeah. One what you call as milligram is 1000 micrograms. So let our happiness be multiplied 1000 times. So let this be multiplied 1000 times. So this will give me a concentration of 200 micrograms per ml, right? There you are, it's done. So if you have a tyrosinase solution whose absorption at 280 nanometer is 0.50, it is 0.2 milligram per ml or 200 microgram per ml. Stay tuned with me, Professor Girish Kukreja, for more in microbiology, biotechnology and biochemistry. Thank you so much.